So today's presentation is being recorded. I'd like to thank everybody for joining us this morning. And with that, I would like to turn it over to Christina Rohina, who will present today on Thunder Accelerated. Christina, the presentation's all yours.
Thanks, Dan. Thank you again, everyone, for joining today's talk on Thunder Accelerated. Like Dan said, my name is Christina Rohina. I'm the supervisor for the Widefield applications team, and I cover Southern California. If you have any questions, feel free put them in the Q&A, and we will get to them at the end of the presentation. In this talk today, I'm going to focus how our family of Thunder imagers, and in combination with Aivia, can allow you to decode biology in real time. And with that, we'll get started. So our Thunder imager family specifically allows the user to reduce the time to result, extend the range of your applications, and increase the relevance of the data that you're generating with the images that you can do with Thunder. Out-of-focus blur masks wanted information in traditional wide field microscopy. This is illustrated by this HeLa spheroid here shown in the background, which is around 200 microns thick. You can see how that out-of-focus blur really masks the details that we really want to observe and maybe analyze out of this image. This makes traditional conventional wide field techniques a bit challenging to use for imaging of samples of this kind. However, with Thunder, it allows you to extend the range of your applications by removing that out-of-focus blur, making thick samples now accessible for analysis. This is also better illustrated by this image of this organoid cluster, which is over 100 microns thick. Thunder does allow you to now visualize the individual cells of this large organoid cluster. This image actually took 469 Z-planes to acquire, but you can see how much detail is actually preserved in the image when you process the image with Thunder. Thunder additionally makes thick samples accessible for 3D analysis. This is an example of a fixed gonad of C. elegans, and you can see as the video goes back and forth between the raw image and the processed Thunder image, how much detail is actually revealed after you process the image with Thunder. And now you can actually use 3D analysis to better quantify the data in this image. This is illustrated by the following image. If you select a region of interest, right, of this fixed gonad, and then attempt to do analysis, what you can see is that if you use the raw image here of that area of interest, you actually miss a lot of data within this image because of the blurriness of traditional wide field imaging, right? So you don't get access to the full data of your image itself. If instead you were to use the Thunder data, you can now see that this reveals the complete volume of the image and is now accessible for data analysis. This, again, increases the relevance of your work by increasing the statistical significance and also increasing the physiological relevance of your work. It does this through background independent quantification and allowing you to apply minimal light dosage to your sample with the use of LED illumination. So what is Thunder itself? Right? Thunder is an optodigital method that removes out-of-focus blur found in traditional wide field imaging in real time. It is actually fully integrated into the system's data stream, and it automatically takes all the relevant optical parameters of your system into account. If we dig a little deeper into how these Thunder methods work, I can better illustrate this with this graphic here. So in traditional wide field microscopy, an image is actually created by the convolution of this point spread function. And you can see that in wide field imaging, we're capturing not only the in-focus light, but also all of this out-of-focus light. This results in what looks like a pretty blurry image here. The technology behind Thunder, which is instant computational clearing, detects this out-of-focus signal and deletes it from the point spread function, resulting in a much more elongated point spread function, and then revealing essentially much better contrast in your image and allowing you to detect the structures of interest in the image. Now, instant computational clearing can be applied to 2D images, meaning you do not have to acquire 3D Z-stats to be able to process your images with Thunder. However, if you do have 3D images, we have the capacity to combine Thunder with deconvolution methods that we have developed for our Lightning technology. So we have two different sort of flavors of this: large volume computational clearing and small volume computational clearing. Each one more dedicated to either thick specimens or thinner specimens. With large volume computational clearing, we actually use the advantage of instant computational clearing or Thunder to remove that out of focus blur. And then to the resulting image, we then use our decision mask technology, which is based on our 3D deconvolution from our Lightning systems, to then clear up much better our point spread function and reducing that residual out of focus blur, and now revealing and enhancing much higher signal to noise levels in your image. Instead, if you have thinner samples, we recommend this small volume computational clearing. This small volume computational clearing again uses our deconvolution, but it does this first. And it does use all of the optical parameters of your microscope optics, the excitation and emission wavelengths, as well as your emission media, embedding media, to process your image. And then this can be further processed with our instant computational clearing technology behind Thunder to really reveal much finer structures in your image and allow you to now quantify things that were masked by the blur in your Widefield image. I mentioned that for our deconvolution, Leica uses our decision mask technology, known as Lightning, as a standard approach really in all of the Thunder processing that includes deconvolution. So what it really is a decision mask. Decision mask is a matrix that detects first all of the high signal to level noise and the low signal to level noise, not only on all the pixels within a 3D stack, but even within single voxels in a single image. So this is shown in the image here, and you can see how a kernel is moving across the image, detecting the noise levels throughout the areas in the image. With Thunder, again, you can get reproducible quantification that is background independent. So this is shown in this example here. We compare a raw image to our Thunder processed image, and we draw this region of interest, this line across the same region in both images, right? And what is obvious here, if you look at the line profile of that line or that region of interest, in the raw image, the peak signal intensities are going up with the background. If you want to do any comparison here, this actually makes it impossible to run any data analysis for comparing the signal intensities here, because it is masked by the background of the image. When you Thunder your image, now you remove the background, but you preserve, right, the structure and relative intensity of these peaks, making it now accessible to be quantified and compared. Thereby, you are now increasing, right, your statistical significance by generating more data. This is also illustrated by the image in the background here. This is a mouse whole mount retina, and by scanning it with our Widefield systems, you're able to do this incredibly fast, process them with Thunder, and then they are ready for automated analysis, allowing you to generate much more meaningful data. We've had some advances as well in the past few years with Thunder, and in particular in the past year, we have released Thunder Live. Thunder Live is really exciting because it actually has the potential to radically simplify your workflow. When you're talking about processing your images, what traditionally happens is you're going to take your image and then you're going to go and process it. And you set your parameters for the processing. And either you've already determined this empirically by having to do this multiple times, or this is just the first time you're starting, and you set the parameters in some sort of informed way. But you don't really know what the effect of that processing is going to be until you actually do it. And then you may have to go back and then tweak those parameters to better fine-tune, right, the processing that you're doing on your image. With Thunder Live, however, you can now get computationally cleared images directly in your live preview, thereby reducing the time to results, to optimal results with Thunder Live's immediate visual feedback. So I'll give you an example here. In this video, you can see a preview of a kidney section. You're looking at the proximal tubule borders here, and you can see that we have the Thunder settings here. And as you turn on Thunder Live, you can see the effects of the processing and the effects of actually tinkering with those parameters, what that effect is going to have on your image. By lowering the feature scale right now, what's happening is that you have better access and better visualization of these proximal tubule borders. And as you scan or go through your image in Z, it'll allow you to really easily set those Z boundaries for Z-stack acquisition, for example. This makes a huge difference when you're comparing your image, say traditional Widefield methods. If the image is really blurry, setting that Z-stack can sometimes be really, really challenging. And this really allows you to do it in real time, get really precise, and get a much better idea of what your processed image is going to be at the end and save you a lot of time. So you're going to intuitively and quickly find the best Thunder parameters on the fly where you're still in the live window. Thunder Imagers also, in combination with a lot of other modules, can get you more data with less effort. So in combination with LAS Navigator, this allows for the use of a variety of different templates and allows you to use much higher format. Instead of just slides, say, or petri dishes, you could use now multi-well plates, load these templates, and really generate large amounts of data with all sorts of major sample formats, making it really easy to integrate into your workflow. Similarly, how can Thunder get you more data with less effort? You can also scan much larger areas and visualize instantly the computationally cleared image as it's being generated, right? Allowing you to get that much more data with essentially the same level of effort as if you were just taking a regular image. Similarly, you can integrate parts of your workflow like analysis into this automatically so that you don't have to wait until your images are acquired to start processing and analyzing your data. You can fully have this be automated in your workflow, allowing you to get data faster into your images. So I've talked to you a little bit about the Thunder technology. Now we'll talk to you a little bit more about the entire Thunder family. What's really nice about our portfolio is that we really are allowing the customer to suit their application to the type of system that we offer, or really pair up the type of system that we offer to their true application. So here you can see a lot of traditional applications that are used in research, anything from monolayer adherent cells, so like cancer cell lines. You now have more like 3D structures, right? Forming spheroids and neurospheres. Examples shown here in the bottom, like the spheroid with these cortical neurons and this organoid cluster. And then even further, as you move into the 3D and thicker sample type, into whole tissues and actual model organisms. So shown here is a rat brain and a zebrafish as well. And what's really neat is that we actually have a system that is suited for each one of those different applications, from monolayers all the way up to model organisms. So Thunder Imagers really allow you to reveal the detail, even on thick samples. This is an overview of our portfolio of Thunder Imagers. We cover everything from high-end fluorescent stereo microscopes, high-end upright microscopes, and our high-end inverted systems. And I'll go through each one of these and some examples on each one. But we do then are able to offer powerful system solutions, right, for all of your core life science applications. Today, I'm going to start with our Thunder Imager model organism. So this is based on our stereo microscope platform, our M205 fluorescent microscope. And the areas of application that it's really applicable is in terms of the visualization of model organisms themselves. So anything like mouse embryos, zebrafish, C. elegans, Drosophila, it's really typical for people to be using fluorescent stereos, right? And routinely, to get better insight into human organism, model organisms are invaluable tools for us to be able to learn about them. So this model organism is shown here, based on that M205 fluorescent stereo, can really be customized to suit any of your needs in terms of visualization of model organisms, right? This system can be customized to suit, again, your particular needs, and even the addition of motorized stages and environmental control if you choose to do live imaging of your whole organisms. So what is your advantage, really? What are your benefits with our model organism? With our model organism, you're able to, again, extend your range of application by acquiring really rapidly blur-free images and showing the fine detail even deep within thick organisms. This is an example of zebrafish sorting. And you can see that you can zoom in to different areas of the zebrafish, and in the thundered image, you can really reveal the vasculature of the fish really easily with thunder. You can get higher relevance, right, by keeping even those large model organisms under physiological conditions during your imaging, again, so you can add environmental control to these systems as well. And you can simplify the handling for much more efficiency. With our model organism, you can reduce the time to result, and we have a workflow example here with C. elegans. So traditionally, if you're doing C. elegans work, a lot of times that's going to involve some sort of picking, right? This can be done in Widefield or phase or DIC. Then there would be some sort of flow sorting to find the correct one of these worms that you need to analyze. And then you move to a higher end system to do the analysis by doing high resolution imaging. And traditionally, in the old sort of workflow, this may require multiple systems to get the job done. So you may need a, basic routine stereoscope to do the picking and transfer your samples over to a fluorescence scope so you can actually do this fluorescence sorting, and then transfer them again to a higher end fluorescent stereo microscope so that you can do the high-end image analysis that you need to do. With our Thunder model organism, there's absolutely no transfer steps. Therefore, you get much quicker to your results. I have a really neat example here of a workflow example of a brain slice imaging on a model organism. And so you can see that initially we get a really fast overview of the sample itself using Navigator. So we're doing this in Widefield to essentially find the area or the sample in our slide. Once we do that, we can then quickly outline our region of interest that we want to image, zoom, and acquire that in fluorescence as well. You can see here how we're outlining the brain slice. And then we just hit start to acquire the fluorescent image. Now, Thunder is being done at the same time. Here, we zoom in to increase the number of fields and increase the magnification and resolution here. And you can see when you compare the raw image, so we're taking a Z-stack here, to the thundered image, how much detail is actually revealed when you look at the processed image. So you can see now all of the dendritic spines of these neurons that were really masked by the blur in the traditional Widefield image. And this was all done without you having to move your sample to another system, just really easily in minutes get you from identifying your sample to really high quality images. Next, I'll show you just a couple of examples of some applications that are really cool that have been done with our model organism. My first example is this cleared mouse embryo that is expressing stable M cherry for its vasculature. And you can see in the left-hand side how you can see blood vessels in the image, right? But if you were trying to do any sort of tracing of these, you would not be able to follow these smaller offshoots from these blood vessels at all in the original blurry image, right? When you process the image with Thunder, however, you can now really easily reveal the detail of each of these blood vessels, and tracing of them is actually enabled. This next example, I just think it's pretty cool. It's actually a 300 micron section of a mouse lung. This was actually taken in plastic here. And you can see that if you're trying to visualize the blood vessels, you can pretty much see them here in the raw image. But if you really want to look at the muscle cells that are surrounding these blood vessels, that's really obscured, right? by this out of focus blur seen here. When you process the image with Thunder, now you can easily detect those smooth muscle cells and be able to adequately analyze your image. Finally, as I mentioned before, you can also, have environmental control of your samples in this model organism. And you can see here with this video of a zebrafish that is stably expressing M-cherry tagged hematopoietic progenitor cells, that in this processed image, you can really easily see single blood cells, right, going through the vasculature of this fish. I was illustrating how Thunder can really allow you to visualize things that are really masked by this out of focus blur. And this is really nicely illustrated by this video of the blood cells, single blood cells being visualized here within the zebrafish animal itself. So I've gone over now our model organism or stereo system. I'll move on to our family of upright Thunder imagers. So these are our tissue imagers themselves. So we have two different flavors, our Thunder Imager 3D tissue that allows full motorized Z control, and then our Thunder Imager tissue for 2D image acquisition. Both of these are based on Leica's intelligent automated upright microscopes, our DM4 and DM6B line of microscopes. So you have now access to fully motorized tissue imaging with instant computational query. As I mentioned before, the use of Thunder or ICC is able to be done on 2D images. So you don't have to acquire 3D Z stacks, thereby saving time and preserving your sample. What's really nice now is that we actually have a new and improved line of Thunder Imager tissue systems with our DM6B in combination with Synapse. What this means is that we are now able to trigger our peripherals in our upright systems, allowing you to speed up your acquisition by even more than 69%. So I'm going to show you an example here of a multicolor slide imaging. On the left is going to be our older systems, non-triggered systems, and on the right, you'll see the system in combination with Synapse. So you'll be able to see fast scanning of large areas of your tissue and getting that contextual information much faster and reliably than you did before. And you can see in the comparison of the speed of these two images, right? Before the one on the left is going to be done, we're going to be able to go through and scan this area essentially more than 3 times. I'm really illustrating the speed of this new upright system. So what is the area of application really for our tissue imagers? That really is tissue, tissue, tissue. You're able to now image tissue sections much quicker than you were before. Our upright systems are also equipped with 4 and 8 slide holders, if you so choose, to also increase the throughput of your tissue acquisition. With our upright tissue imagers, you decrease the time to results. So you have rapid acquisition of blur-free images, even deep within tissue sections, deep within thick tissue sections. You're able to get the fast overview of these whole tissue sections, and you're able to get higher relevance by imaging and analyzing even thick and challenging sections with an easy workflow. So regardless of whether you're investigating, neurite projections, lymph node architecture, or regenerative responses, you can effectively remove that out of focus blur with your Thunder tissue imagers in real time and get maximum ease of use. I have just a few examples of some applications that were done on our tissue imagers. This has to be one of my favorite, I think. This is a YFP tagged neuron mouse brain slice that's also been stained with anti-GFAP in 647. And you can see really dramatically what effect Thunder has on this image. When you see the raw image here, you can see the head of the neurons, but you can't see any of the projections, any of the neurites at all. And really, those GFAP tagged cells are quite difficult to visualize and localize. Like this would be an image that would not be able to be, quantified in any significant way, and it's raw. But Thunder really reveals the detail behind it and allows you to get more relevant information from your images. One of the main applications, right, of our tissue imagers are going to be these tissue scanning So these tau scannings of large areas of tissue. This is an example of a slice of a mouse brain. And you can see how much detail is revealed in the processed side of the image. You can really tell all the different cell populations that are within this mouse brain slice that are really obscured in the raw image. Finally, I have this one last example of a neuromuscular junction. This has to be one of my favorite images just because it looks like just a beautiful flowered tree. Once you process the image with Thunder, you can see here that in the raw image, the neuromuscular junctions are visible, but really none of the details are. And now with the processed image, you can really trace all of the neuromuscular junction and visualize, right, the other tag here in this image, this beta bunker toxin, and really see where those signals are coming from and where in the neuromuscular junction they are located. Now I'll move on to our final member of our upright family, which is our Thunder Imager EM Cryo CLEM. CLEM stands for Correlative Light Electron Microscopy. And this imager really allows users to fit our Thunder technology within their EM CryoCLEM workflow. So this is our upright system, and we have a Cryo 50x.9 numerical aperture objective, as well as a dedicated shuttle system for grid loading within the microscope itself. So what really is the area of application that we're looking at here? So traditionally, when you're looking at EM CryoCLEM, this is a pretty significant long workflow that involves multiple systems, multiple steps. And once you get to the final EM step, you really want to make sure that you have quality samples, right, that are not contaminated, and that are able to visualize your target of interest, thereby really positioning Thunder to this particular application. So you can really use this system to do quality check of your sample. So in CryoCLEM, ice contamination can be a really big issue, and you can use this system to really check whether or not or what areas of your grids have contamination prior to that cryo EM imaging. Similarly, you can use this to get much better and much more precise identification of target structures for EM images. So what are the benefits of the Thunder CryoCLEM? Essentially, you're going to avoid the blur of a Widefield system when imaging these thicker samples. So here is an example of these cells here, and you can see how different it looks when you process this with Thunder, allowing you to identify really precisely the target of interest within your image. You're able to get higher relevance because you have higher resolution images, right, due to that 50x.9 cryo objective that comes with the system. And you're able to reduce the time to result by having that better identification of your targets, to visualize them easier, and to also check, the quality of your samples, allowing you to still use a Widefield microscope, which allows you to really take advantage of the speed of Widefield to image your samples here. I have just two examples. I'm going to go over again the same image I just showed you. So these are actual yeast nucleoli that we need to target here. And you can see that in the raw image, the identification of this nucleoli within each cell would have been quite hard. However, with the Thundered image, you can really see the detail of which cell is which by looking at the cell walls here, by visualizing those cell walls, and really much better identification of and outlining of the nucleoli than before. All right. This is my final example of our CryoCLEM, Thunder CryoCLEM. And you can see here in this particular example, we are interested in looking at the trans-Golgi network. So these are HeLa cells that have been stained with F-actin in M-cherry, and then a trans-Golgi marker in green. You can see on the left here, that Golgi network is quite obscured by the blur, whereas here in the image, once you process it with Thunder, they're really easily visible, and you can thereby target what area of the image you want to then further process on the EM cryo workflow. So that really covers our stereo and upright systems. And now we'll just move to our inverted family of systems, which covers both our 2D, 3D assay, and then our live cell imagers as well. So with this particular area of our portfolio, we are allowing you to extend the range of applications, right, to thicker samples and also maintain physiological control of your samples, right, by allowing you to control the live cell environment. Traditional Widefield microscopy is really great at the imaging of, really sort of thinner samples, monolayers of cells, maybe some kidney cell domes. But as you start getting thicker and thicker, the Widefield microscopy becomes a little bit more challenging, again, because of that out of focus blur. However, with our Thunder imagers, it does allow you to extend your range of applications by really being kind of independent to the thickness of your sample as far as is shown here. So really with our inverted systems, you're able to reduce that time to result by upscaling your discoveries. With our 3D assay and live cells imaging systems, you can really access a wide range of challenging 3D samples, anything from spheroids, neurospheres, and tumor spheres, brain or intestine organoids. You can really have sort of a limitless accessibility to the type of samples that you can image. In our advanced cell culture experiments, such as live cell explants, vibrotome sections, and even photomanipulation experiments like TURF and FRET, which was the example that was shown here on the left. All of these types of applications are accessible with our inverted line of Thunder imagers. When it comes to the inverted systems, again, we have first our most basic system, our Thunder Imager 3D assay. We also have our Thunder Imager live cell, now allowing full control, right, of your environmental conditions, both temperature and CO2 conditions, with the fully motorized system here. And our newest member of our inverted Thunder family, our Thunder Imager Nano. So this is now further adding to your CLEM workflow solution, but now looking more at live cell CLEM workflow solutions. So really this allows you, if you're doing any sort of live cell cryo CLEM applications, to really monitor your particular sample, right, for the event that you're really looking for to capture further down in that cryo workflow. This instrument itself, right, comes with this sampling chamber that allows you to better transfer any samples that you're doing further down into your workflow. So really allowing you to capture critical moments or critical events that are happening within your sample prior to freezing. If you're interested at all in learning a little bit more information about our Thunder Imager Nano, we do have a webinar that is in LabRoots that explains a little bit more about how the Thunder Imager Nano fits into your nano workflow. So now I'm just going to give you some examples of some applications that were done on our inverted systems. This is a really cool example. I think it's very common for researchers to do work on, monolayers of cells. These are cultured viral cells that were stained for vimentin and a mitochondrial marker. And you can see that in the raw image, you can see that there's structures there. You can see that the mitochondria are labeled, but you can't really do any sort of segmentation out of that image. You can't do really any sort of detailed analysis because of that blur that is seen in the image. Now with the processed image, you can really see the details of each of the mitochondria and of the intermediate filaments labeled by vimentin here. That was really impossible to observe in the raw image. Another example of some applications that we've done with the inverted system is this 3D reconstruction of a human pancreatic islet. And you can see here on the left, this sample is stained with multiple markers to identify pro-inflammatory cytokines here, so different subsets of types of cells. And in the left, you can see that, there's really cells in there and that there's different markers but you can't tell what cell expresses what at all. And if you wanted to do quantification in terms of the number of cells in this image, it would be particularly hard to do so. However, on the Thunder processed image, you can clearly see the different subtypes of labeled cells. You can clearly make out each of the individual nuclei, making it much easier for this image to be segmented and analyzed further. My final example for our inverted systems is this video of our calcium imaging on an Arabidopsis thaliana seedling. You can see how with Thunder processing, you can capture all these bursts of calcium, not only in these kind of denser areas, but also in these much smaller structures down here that would have been really difficult to visualize, right, in traditional Widefield imaging. So this is a really neat example of some of the live cell work that you can do on our inverted systems. Finally, I will speak to you a little bit about Thunder in combination with Aivia and how that can increase your data quality. So Aivia is our AI image analysis software that makes advanced data analysis accessible really for all biologists. Aivia's AI-powered analysis capabilities essentially leverage your expertise as a biologist to really generate robust and reproducible segmentation results. You essentially train your software to segment your data and analyze your data. And I have a really neat example here using the same image of the human pancreatic islet. You essentially can use an algorithm like the pixel classifier to train the software to segment, say, nuclei from your image. You essentially paint on your image the areas of interest and the areas of background, and then the software itself can really easily segment now your image into individual nuclei and get whatever type of data analysis you need to get from your image. This is better illustrated in the whole islet here, as you can see how easily you're able to segment out each of the nuclei of that image. So with Thunder and Aivia, you really can't increase your data quality. You can accurately segment your image and open up new experimental options for live cell imaging. These are just a few examples of some applications that were done of Thunder in combination with Aivia. Here you have an intestine organoid. This was labeled with SM fish. And you can see that even though this is a very dense, organoid, you can see that with Aivia, you can really finely segment each of the individual cells of this organoid that may have been difficult to do with traditional segmentation based on thresholding. Another really common example of something you can do with Aivia, right? You're counting nuclei again, so segmenting nuclei. But what you can see in this image is that, so this is a monolayer of breast cancer cells, but this is quite dense, right? They're pretty, not overgrown, but they're pretty confluent. Sometimes this can be quite difficult to adequately segment, but you can see how well of a job Aivia did in not only segmenting it, but also illustrating the different types of populations that may be in this region of interest. Finally, I have here, this is essentially the same image I showed earlier of the YFP tagged mouse brain slice with the GFAP tagged cells. And you can see here, this was actually processed with our 3D neuron analysis in Aivia. And you can see how you're able to track all of the neurites from the neurons here really easily and really quite quickly with the Aivia software in combination with Thunder, because this would have been pretty difficult to do on the raw image itself. And so with that, I'm going to go over just some resources that we have available for you if you want to get more information about Thunder. We have a variety of different white papers that we have generated, everything from, digging deeper into the actual technology of Thunder to actual guides that can guide you through better live cell imaging techniques and fluorescence imaging itself. We have tutorials that are really popular on our YouTube channel, our Leica Microsystems YouTube channel. And we have a variety of different publications that you can access that are focusing on one of the many different members of our Thunder family. Similarly, we have a lot of resources geared towards applications that were done using Thunder. We have case studies, image galleries, different workflows that we are showcasing here in our science lab Thunder imaging section, as well as interviews with different users, webinars, and abstracts of things that have been done with Thunder. And so with that, I really want to thank everyone for attending and for your attention. Again, encourage any questions that you may have. And I also want to let you know if you have not only questions about Thunder, but need any help through contacting someone here at Leica, feel free to contact either myself. Again, I'm the supervisor of the Widefield applications team and support Southern California. But also if you need to be put into contact with sort of the right person in your area, you can also contact Jen Lee, who's our marketing manager for Widefield in the Americas. And we also have additional resources, like I mentioned, in our Leica Microsystems tutorial channel on YouTube, and also our remote applications team. And with that, thank you again, and I welcome any questions.
Thanks, Christina. And I would like to encourage anybody who has questions to please use the Q&A function, and we will answer them as they come in. I do have some questions that came in directly. First question, very, very basic key question. If I already have Thunder, can I add Thunder Live to it?
Yes. So actually that's something that can be very easily done. You just need to contact us and we can really easily add Thunder Live to any existing Thunder system.
Great, thanks. And the kind of follow up to that was, what about the possibility of a Thunder offline workstation?
Yes, this has been one of our biggest asks, I think, throughout the years as we have released Thunder. And that is actually a possibility now. You have the ability to get offline Thunder workstations, really freeing up the ability for you to even generate more data and have a dedicated system that's just for image acquisition, and then a secondary system that is geared towards all of your processing needs. That is available now.
Thanks. Also, what do I need to upgrade a Thunder 3D Thunder 3D tissue to the new faster version?
That is a good question. I think there you'd want to definitely contact your account manager, and they can guide you really through what needs to be done to upgrade the system to now the triggered version of our Thunder tissue imagers.
Okay. And Aivia related question, how can I get a trial license for Aivia?
Yes. Again, contact either myself or Jen Lee. We can get you in contact with your local account manager, and they can really facilitate getting you a trial license of Aivia so you can try that out and really have a kind of a more in-depth conversation as to how Aivia is better suited to your particular application needs.
Great. Thank you, Christina. And those are the questions that I've got so far. And I'm not seeing any fresh questions coming in, so I'll just take this opportunity to thank Christina for giving today's presentation. And thanks everybody for joining us. Much appreciated. We appreciate your time, and we look forward to chatting with you in the future. Thanks, and have a wonderful rest of your day.
Thanks, everyone.